So the title of this talk is Receptive Field Size and Scene versus Phase Preference in the Hippocampus and Medial-Temporal Lobe. The authors are Charlotte Leferink, Jordan Cracker, Eva Brunig, Stefan Kohler, Morris Moskovich, and Dirk Bernhard Walter. So this talk will be organized along the sections of navigation specialization within the hippocampus, looking at particular specialization within the anterior-posterior axis of the hippocampus, moving on to the specialization and organization within the medial-temporal lobe, memory-related areas, and then looking at the relationship between population receptive field size and scene specialization within the hippocampus. So first off, talking about navigation within the hippocampus, we know that there is a specialization that's particular to the hippocampus relative to the more perceptual scene activated areas within high-level visual cortex, such as the hippocampus as shown in this study by Haslakis et al. Hippocampus is known for its role in navigation, for its role in decoding the location of an individual within a scene, which was one of the tasks in a virtual reality experiment, versus that of decoding the between the rooms, differentiating between the rooms, which was evident within the parahippocampal cortex. And so given that the hippocampus is well known for its role in encoding and in retrieval of memories, it is an interesting area to investigate population receptive fields and visual activation for specialized features. So we also see that there are lateral effects within the hippocampus, where we know that for the study by Bauman et al., where decoding showing where location within a scene, navigating a scene, effective strategies for retrieval during encoding were found within the right hippocampus, and less effective decoding strategies or retrieval strategies were found in the left hippocampus. So we also wanted to look at not only are there different laterality effects within hippocampus, but we also know that there is a differentiation along the anterior-posterior axis of the hippocampus. And this is evident by a study from Woolett et al., where taxi cab drivers who were trained on navigating London city streets showed a difference in volumetric size, but only within the posterior hippocampus after a significant number of hours of training. And so we also know that within rodent studies, we see that there are different gradient resolution within the dorsal versus the ventral hippocampus, which is homologous to the human, which is on the right here. The human hippocampus for the posterior is homologous to the dorsal, and the anterior is homologous to the ventral in the rat, where the ventral area of the hippocampus in the rodent showed a more a large coarse-grained scale encoding of the spatial environment, whereas the dorsal area showed a more fine-grained representation of the environment. And because of these differences in rats or rodents, we also know from past research exploring this differentiation in humans along the anterior-posterior, where a study by Hirschhorn et al. Showed that activation for navigation between 2 points within Toronto city streets, where participants had to imagine navigating more fine-grained details between 2 location landmarks within Toronto. They found posterior hippocampus activation for this more spatially fine-grained task. And not only do we see that there are differences in the functional for the activation of the hippocampus along the anterior-posterior axis, but we also know that there are different functional connectivity, different functional connectivity within the hippocampus to other areas in the surrounding medial temporal regions and beyond that in the hippocampus, where the anterior hippocampus has been shown to connect through Dalton, connect more to the ventral medial prefrontal cortex, and the posterior hippocampus is connected to the inferior parietal cortex, so more downstream areas in cortex. as well as we know that the posterior hippocampus is connected to the cuneus, the precuneus, the anterior and posterior cingulate cortex, as well as I said, the inferior parietal cortex and also parts of the thalamus. And not only do we see that there are different functional areas, connectivity within the hippocampus, but and different specialization, functional navigation abilities in the hippocampus, but we also note that other past studies have shown that the apex of feature discrimination occurs within the medial temporal areas, such as the perirhinal cortex and the entorhinal cortex. In the study by Benson and Saxida in 2007, they noted that for items that are highly similar, the discrimination ability is decreased relative to controls when the perirhinal cortex is no longer available for the task. So given that we have these different medial temporal areas with different functional specificity, we wanted to include them in our analysis of in the hippocampus. So here we looked at the segmentation of the medial temporal area, such as the entorhinal cortex, which was segmented into the anterior lateral and the posterior medial, as shown here in the red and the green in the middle figure. We looked at the gradient along the anterior posterior axis of the hippocampus, which was segmented using Jordan de Cracker and Stefan Kohler geodesic segmentation process, which allowed us to obtain a gradient from 0 to 1 along the anterior posterior axis of the hippocampus unfurled, so accounting for the curvature and sort of flattening the hippocampus out so that the uncus, or the anterior part, most anterior part of the hippocampus, was the most anterior part on our gradient of 0 to 1. So we use the data from the Human Connectome project, 181 participants, of which we used 157 of the participants. We also used the data set from Noah Benson and all of the retinotopic parameter fitted using Kendrick K's 2013 spatial summation, compressive summation, spatial summation algorithm. We used as I said, the 7 Tesla from the Human Connectome project included a retinotopy experiment, which was comprised of images, composite images, such as those here shown in A, which were of objects, faces, places, and body parts. And this composite image was designed so that it would stimulate areas within high-level visual cortex, not just the checkerboard patterns, which wouldn't wouldn't activate the high-level visual areas as much as these specialized features would. And we also looked, so the retinotopy experiment showed these composite images moving on the background while participants fixated, and only a part of this composite image was shown to participants at a time. And this was to occlude parts of the image in order to isolate visual cortex that was activated according to the image. So in figure B, you can see which parts of the image, what type of stimuli were occluding the composite image at a time. So there was a wedge, only the white part of the wedge was showing the composite image at a time. So it would be a wedge rotating clockwise or counterclockwise, a ring that was in the center of the screen expanding or contracting, as well as a bar that was translating left to right or diagonally across the screen. And from the fitted parameters, so each population of receptive fields within each voxel was fit, again, according to the compressive spatial summation algorithm, we obtained the parameters of basically the polar coordinates of the location of the receptive field size on the viewing plane, as well as the size of the receptive field. So how much of the viewing plane was being processed by a given population of voxels, population of neurons, to give us a population receptive field. And using these parameters, we were able to map out the part of the viewing plane representation within each of these areas, the medial temporal regions, including the hippocampus. So the 2 areas that showed a contralateral hemifield bias, which we know is characteristic of early visual cortex, or retinotopic, parameters, we showed that in parahippocampal cortex, there's a contralateral hemifield bias, as well as, which has been shown before by Silson and all, but we also showed that there's a contralateral hemifield bias within perirhinal cortex. We did not find a contralateral hemifield bias within the entorhinal cortex, neither the posterior, medial, or the anterior lateral parts of the entorhinal cortex. We also looked at the hippocampus, which has also been looked at by Silson et al. In 2021. And we, using our geodesic gradient of the hippocampus, we showed that we parceled it into 5 equal parts. And we showed that and we mapped out the contralateral, or the representation of the viewing plane in each of these parts bilaterally. So on the left graph is the left hemisphere, and the right graph is the right hemisphere. And on the left graph, in the middle 2 anterior segments, we see a contralateral hemifield bias, and we do not find the contralateral hemifield bias on the right hemisphere. And this is different from what Silson et al. Found, where they found bilateral hemifield bias within the middle hippocampus, and we only find it in the left middle anterior, though they do find only the right hemisphere has and contralateral hemifield bias in the anterior region. So there are lateral effects of contralateral hemifield bias across both of our mappings of the viewing plane within hippocampus along different parts of it. So in addition to mapping out the retinotopy parameters for the viewing plane, we also wanted to see the representation of the size of receptive fields within these, so medial temporal regions. So we find in the parahippocampal cortex larger receptive field size on average relative to all the other areas of interest in our study. And this is expected given we know that this has been shown in past research by Caro et al. But we also find that within the hippocampus along the gradient, there is an increase in receptive field size going from posterior to anterior. And we also find that in the perirhinal cortex, there is a large distribution of receptive field size, though they do tend to be more smaller towards the smaller size of receptive field. And so then using this mapping a comparison of the scene versus face activation. So again, looking at this feature specialization, but here we use the data from the 3 Tesla Human Connectome Project experiment from the NBAC task, using the face and place images as our activation when participants are looking at these images, together with the help of my colleague Claudia Damiano. We together, this is the average showing here of the scene versus face contrast coefficient. So the activation for scenes is shown in the red color here, especially in the parahippocampal cortex, which is shown in this bilateral. So the right hemisphere on the right, left on the left here in this image. And we see that there's an increase or a gradient within the hippocampus, so more evident within the right hemisphere, going from less scene activation to greater scene activation along the anterior-posterior gradient. And we also note that in the perirhinal cortex, there is greater face activation than scene activation. And these results align nicely with the size of receptive field that we found. So large receptive field size in parahippocampal cortex aligns well with the greater scene activation within parahippocampal cortex. And the gradient going from smaller receptive field size to larger receptive field size aligns nicely with the scene activation within the hippocampus, where less scene activation in posterior and greater scene activation in anterior aligns with the larger receptive field sizes in anterior hippocampus. And these effects are also shown in a mixed effects model statistical test, where there is a significant increase from posterior to anterior in the hippocampus for both the size of receptive field and the coefficient of scene versus face activation. And this relationship is also significant, where larger receptive field size is possibly related to more scene versus face activation within the hippocampus, along again, measuring this along the anterior-posterior gradient. And so our takeaway from our findings, though we see this population receptive field size and specialization for scenes is evident in both the perceptual and the memory processing areas, such as those shown as the hippocampus, as well as the more perceptual parahippocampal cortex. And so we note that larger population receptive field sizes process scenes across the range of function. As I mentioned in the beginning, there's different types, different navigation abilities, different spatial representations, as well as the more perceptual areas. And yet, though they have different functional requirements or objectives, we note that they maintain the retinotopic properties, such as the contralateral hemifield bias that we see in early visual cortex. And we note that these retinotopic parameters showing that the representations in memory of the scene activation aligns with the perceptual representations, where larger receptive field size aligns with greater scene activation. And this also suggests that there is a similar spatial encoding representation across the perceptual and memory related areas, or spatiotopic encoding and indexing of the features in physical space, also represented similarly, analogously within the perceptual and the memory related areas. So I'd like to thank Jordan DeCracker, Stefan Kohler, and Eva Brunick, and Morris Moskowitz, the collaborators on this project, for sharing their expertise on memory related areas. And thank you to Noah Benson for his assistance with the retinotopic data set and the analysis, dataset analysis. And would like again to thank Claudia Damiano and Stefan Kohler students for their assistance with the segmentation of the entorhinal cortex. Thank you.